Okay, thank you very much. It is indeed a great privilege to be here. Thank you very much for inviting me. So I took the title of this conference and with a question mark, and I want to say a few things about the concepts that are involved, that we're using, and to tie it up with the kind of thing that we heard, and to sort of present some kind of I don't know, a little bit historical, a little bit conceptual, a little bit of a mixed discussion. So, first of all, I would like to dedicate this lecture to my late friend, Louisa Hirschbein, who was a French biologist and epigeneticist. So, to her memory. Okay. So, when we're talking we're using the term acquired traits. What exactly do we mean? It's not such an innocent word. It's not such an innocent phrase. We know what we're talking about. It could be useful to sort of spell it out. And then, what is the relationship between plasticity, phenotypic accommodation, and acquired characters or acquired traits? So I want to tie up this old term acquired characters and tie it up with more modern terms that we're using. And then ask what is epigenetics, because I think that there is a lot of discussion about epigenetics, not here in this room. I think we more or less know what we're talking about, but in general in the public. And there is a lot of, and some of the discussion is due to the fact that people are using different conceptions of epigenetics. So I think it would be good to say that what is going on and how actually we use this term. And then I want to talk about inheritance. We're talking about transgenerational inheritance, about transmission of epigenetic information from one generation to the next. So what do we mean when we say this? What kind of model do we have in mind? Do we have something, a model like the model of DNA replication? Do we have a different model, a model of reconstruction? What exactly are we talking about when we're talking about transmission? And so, what is epigenetic inheritance? If we have some kind of model, what is it? What is soft inheritance? What do we mean when we say that patterns of changes in the epigenome are affected by developmental changes? And then we come back to the question of, okay, we heard a lot about many of these topics. So what is the role of epigenetic mechanisms in the inheritance of acquired characters? And if we clarify this concept, can it help us? Can it help us to design better experiments, to have some insights into what we're doing? I think it can help. So I'll start with the question of what are acquired characters. So you know, we use acquired characters to refer to a phenotype that was induced or acquired when conditions change, but it's not usually a passive kind of response. For example, when we break a bone, this is not what we refer to as an acquired characters, but when the bone heals, and let's say even if it is crooked, this is an acquired character. When we're scratched, the scratch itself is not an acquired character, it is the scar that we develop that is the acquired character. So in all the when the term was used historically by most people, including, by the way, by Lamarck, who made the point that it is not passive, the organism has to be involved in the acquisition. The acquisition is happening to somebody, to an organism. It is not passive. And it is dependent on mechanisms within the organism. So this is the first thing that we have to remember. That all living, and indeed we have to remember that all living organisms acquire characters in this way, and because otherwise they would be dead. So, but I want to put a kind of disclaimer here. We like to make very clear distinctions between things. We have we have active acquisitions. It's not always so simple. When we're thinking about, for example, the acquisition of the microbiome, there is also a passive element to it. So, but what I want to say is that usually when we're talking about acquiring a character, we're talking about some kind of involvement of mechanism within the organism, active mechanisms. Today, when we're talking about the acquisition of a new character, we're talking about it in terms of developmental plasticity, of having plasticity. And the person who wrote most deeply about plasticity from an evolutionary perspective and from a developmental perspective is, I think, Mary Jane West-Eberhard. And she defined plasticity in the following way: the ability of an organism to react to an internal or external environmental input with a change in form, state, movement, or rate of activity. It may not be adaptive, although very often it at least enables the organism to cope. It may not be optimal. And plasticity is sometimes defined as the ability of a phenotype associated with a single genotype to produce more than one continuously variable alternative form of morphology, physiology, and/or behavior in different environmental circumstances. It refers to all sorts of environmental-induced phenotypic variations. Plasticity involves responses that are reversible, irreversible, adaptive, non-adaptive, active, and passive. As I said, most have some active element, and continuously and discontinuously variable. That's her definition. Very, very wide, and deliberately very wide. Afterwards, she sort of qualifies it in 803 pages. And I want to point out that plastic changes include maturational and non-maturational developmental responses, learned responses, something that is mediated through the nervous system, and also open-ended responses which accommodate novelty, which are something that we cannot say this was part of the developmental program of the organism. It's something that the organism never was exposed to, neither in its own developmental history, nor in the history of the lineage. So there are all kinds of new things that happen, and nevertheless, in spite of the fact that we don't have a program for them, we manage them. And it's very important to understand what is going on, what kind of means do organisms use. So West-Eberhard developed the concept of phenotypic accommodation, which is the set of mechanisms that adjust the organism as a whole to developmental change. And they're mediated through general biological properties such as mechanical flexibility, that is some kind of the stuff, the biological stuff that we're made of is very, very important. Multiplicity of partially overlapping regulatory elements, and through developmental processes such as exploration and selective stabilization at the cellular, physiological, and behavioral levels. And these processes lead to correlated changes in various aspects of the phenotype. This is a very general statement, and one of the things that we have to do is put some flesh on these bare bones. And I think it's one of the great challenges that, with which we're all faced. However, what I want to say, just a few words about plasticity. That plasticity can take, as West-Eberhard pointed out, and many others pointed out, can take many forms. It can lead to an either-or phenotype. So, for example, you can be a male, the colorful fish in the middle, or you can be a female, and this depends on the social environment. If the social environment is such that there are a lot of females around, one of the females will become a male. This is plastic phenotype. It is completely dependent on the environment. The same is true about caste determination in bees. The queen bee will become a queen bee because it eats royal jelly. If it's not fed royal jelly, it will develop into a worker. So this is it's called polyphenism. It is the kind of discrete kind of phenotypes that are based on the same genotype. We can also have a continuous reaction norms, graded phenotype that depend on various environmental cues. So it can be a very light mouse, it can be a slightly darker mouse, very dark mouse, and so on. And the same is true with height. And we have this kind of phenomenon has been known for ages. People have been studying it in the 19th century. We have been studying in plants for many, many years, and it has been used also by French Lamarckists quite a lot. The effect on for example, on plant phenotypes, the graded effects. We also have stress-induced plasticity. So here is a very famous example of a goat that was born without its hind legs, and it learned to walk on its back legs, and it learned it because somebody helped it. She was lucky enough to be born in a farm of a vet, and when she died, unfortunately, of an accident, the post-mortem showed that there were a lot of changes in the muscles, in the bones, in the innervation, in every aspect of the phenotype that was related to locomotion. And this is not very surprising, of course, but we have to remember that this very adaptive phenotype was developed it was not based on any program, on any genetic program, because goats don't walk on their hind legs. This is highly abnormal. Nevertheless, the goat managed. So we can do something which is beyond the normal envelope of programs, of what we call programs, which is also a problematic word. Another thing that we know is that we can stretch plasticity across generations. So plasticity does not only have a spatial morphological here and now aspect, but we can also think about plasticity as for how long can we stretch this change. And sometimes we can stretch it through generations, sometimes through more than one generation. So we can think about epigenetic inheritance or transgenerational effects or intergenerational effect as a temporally extended plasticity. Now, okay, so this is, and it is clear that when we're talking about acquired characters, we're talking really about what today people are talking about when they're talking about plastic phenotypes, and we can qualify the type of acquired characters in a more precise way. Now, when we're talking about epigenetics, we've already heard that epigenetics is the branch of biology which studies the causal interactions between genes and their products, which bring the phenotype into being. And we saw the upper picture and we saw the down picture, and really what Waddington was talking about was that epigenetics is the study of the interface between the genotype and the phenotype. So it is the network that we're seeing here, the network of interactions of the strings that create a network. And it is the property of this network which is very, very important, because the property of this network, the regulatory interactions, are those that underlie what we call plasticity on the one hand and canalization on the other hand. And therefore he was very interested in this network. He called it the epigenotype. He talked about it in terms of network before gene networks and before Jacob and Monod. But after Jacob and Monod, of course, Jacob and Monod gave a very precise meaning to what is meant by gene regulation, which he couldn't at the time give. Now, it's important to understand that terms evolve, and the way that we're using epigenetics today is not the same way that Waddington used it. Waddington had this general view. When we're using epigenetics today, we have a more confined view, and we're talking, and I gave here two definitions. Another very useful definition by Bird was presented in the first lecture. I'm giving two just, but the first is Marcus Pembury, The Science of Enduring Changes in the Pattern of Gene Activity During Embryo Development and Beyond that do not involve alteration of the DNA sequence. These changes occur in response to conditions within the embryo and more generally. So he's saying, look, everything that has to do with changes in gene expression, which go beyond the level of changes in the DNA sequence, is what epigenetics is about. But he's saying here a very important word, and that is enduring changes. Not any change in gene expression, but the change that is enduring. And this is the modern way in which we use the term epigenetics. We need to have some mechanism that lead to the maintenance of environmentally or developmentally induced changes. If we don't have this maintenance mechanisms, we're not talking about epigenetic mechanism. There is a lot of noise, there is a lot of gene transcription that is not maintained through dedicated mechanisms like that. And when it isn't, we're not talking about epigenetic mechanism. And that is why Marion Lamb and I, when we were giving a definition, we said epigenetics is the study of developmental processes in both prokaryotes and eukaryotes, because there are processes, epigenetic processes in prokaryotes that lead to persistent self-maintaining changes in the states of organism, the components of organism or the lineage of organism. And the self-containing, there is some type of autocatalysis involved, always. Otherwise, we're not talking about epigenetic mechanism. And I think that there is a general consensus that this is the kind of sense in which we talk about epigenetics today. And if we remember it, I think that a lot of the non-productive discussion that is going on in the public or among scientists will go away. Now, we're also talking about epigenetic mechanisms. And the notion of epigenetic mechanisms is also something that changed, because at first, when people were talking about epigenetics in the modern sense, they were talking about DNA methylation. Because DNA methylation was the first epigenetic mechanism that was discovered. So when Holliday is describing epigenetic mechanisms, he's talking about DNA methylation. Later on, chromatin modifications came into play. Then people said, well, it's not just that, it's also self-maintaining loops. Then people added also prions. And then people also added small RNAs once they were discovered to be not only part of the regulatory system of the cell, but part of the inheritance and the memory system. So this also has changed, and I think that our present concept of epigenetics and epigenetic mechanism include a host of mechanisms which are very often interacting with each other, especially when we're thinking about the RNA mediated systems and the chromatin marks, which include DNA methylation, histone modifications, and so on. But also the others, because it could well be that some of the, for example, proteins that are produced are prion proteins, and, the sky's the limit. Okay. So now, as you know, there was quite a lot of resistance to the idea for quite a long time that developmentally induced changes can be inherited. And Ernst Mayr said in 1980 that it was perhaps the greatest contribution of the young science of genetics to show that soft inheritance does not exist. Soft inheritance is a term that was invented by Darlington and later used by Ernst Mayr to describe a gradual change of the hereditary material that was assumed to be DNA, brought about by use or disuse, by some internal progressive tendencies, or through direct effect of the environment. The definition is from Mayr. So, for him, it was very important that you can make a distinction between heredity and development. And for him, this distinction between heredity and development is what introduced a unification into biology, what made evolutionary theory truly unified and truly neo-Darwinian, not Darwinian, neo-Darwinian, because Darwin, as we have learned today, did not exclude soft inheritance. On the contrary, he was the one who invented the most detailed model of soft inheritance in the 19th century, but it was excluded by the neo-Darwinians. So at the moment, the question is, okay, do we have soft inheritance? What is soft inheritance? And when we're talking about epigenetic inheritance, are we talking about soft inheritance? So let's define epigenetic inheritance. So first of all, not everything that when a mechanism of epigenetic is involved, it doesn't have to lead to inheritance. For example, there are epigenetic mechanisms in brain cells that do not divide. Nevertheless, there is self-maintenance, there is the autocatalytic processes involved that are responsible for cell memory without division, without cell division. When cell division is involved, then we're talking about cell heredity and about epigenetic inheritance. So we're talking about phenotypic variation that do not stand for variation in DNA-based sequences that are transmitted to subsequent generations of cells or organisms. And we use it in various ways, broad and narrow. And this is also one of the causes of, I think, lack of clarity, because sometimes people mean the broad term, sometimes people mean the more narrow cellular molecular term. But I think that if we qualify what we're talking about, we are very clear about it. I don't think anybody had a problem understanding what people were talking about today. So what is important, that if we're looking at eukaryotic cells, and this is what we're focusing on most of the time here, is we're talking about transmission mechanisms that go through mitosis, epigenetic mechanisms that allow the transmission, that allow epigenetic inheritance through mitosis, meiosis, or even from in multicellular organisms from soma to soma. And it sometimes also involves complex mechanism of niche construction in this case, and in the soma to soma case, we're talking about a broad concept of epigenetic inheritance. Okay, so what do we mean by inheritance? We are all sort of used to thinking about DNA replication. And this is our model of inheritance of biological stuff. But I think this is the wrong kind of, it's wrong to base on our notion of epigenetic inheritance on DNA replication. Let us think what replication involves. Replication is a process that is resulting in similarity between ancestor and descendant based on some process of copying. And this copying is indifferent to the functional significance of the copied entity. And the paradigmatic examples of this are DNA replication, photocopying, and blind imitation. These are paradigmatic process of copying. But we can also, let's say we have phenotype A in generation 1 and phenotype A in generation 2. Now, we want to follow, so there is some kind of transmission must have happened, obviously, especially if this is not a law of nature, but if it is a variation that is transmitted from one generation to the next. So if this is the case, do we really have to see that, for example, we're looking at methylation pattern in generation A1 and methylation pattern in generation 2. Do we have to see the same pattern of methylation throughout the development in meiosis, in early embryogenesis, in slightly later embryogenesis, and so on? I don't think so. What is important is that the DNA methylation is reconstructed with fidelity. And this is a process of reconstruction. So it could be the case that DNA methylation is completely erased during meiosis. But some other components of epigenetic systems, for example, small RNAs, are in place. And these small RNAs, in later development, lead to the reconstruction of this particular methylation pattern. So we will see the disappearance of the methylation pattern and its reappearance later due to something that is indeed being transmitted, but it is not the same thing. And we can think also about something like, for example, it doesn't have to be small RNAs, although they are good candidates. It could be also modifications of histones and so on. So I think that we can have very high fidelity of reconstruction without having a replication process, and this is very likely to be the case with epigenetic marks. And when we're thinking in this way, what we really should focus is again on phenotypic continuity between generations. How is this continuity materialized? How is it established? What is going on? And again, we're going back to the ideas of Lamarck, because Lamarck was very interested in exactly that, in phenotypic continuity and in the things that maintain it and that change it over time. And he had his own ideas about it, but this was the point of departure for him. So I think that we are talking about, so I think that epigenetic inheritance, especially in multicellular organisms, is an auto-regulatory process of reconstruction. Although there may be some elements of replication too, because we do have some elements of replication, for example, with DNA methylation, where methyltransferases are leading to, but you know, it's just part of the story. Okay. So what I want to say is, I think that we can say that if we take these definitions on board, and we take also everything that we have heard here, epigenetic inheritance is necessarily involved in most transgenerational effects. And the reason that it is necessarily involved is that if we're talking about long-term changes, that are something that is maintained over a long time, this maintenance must be based on some autocatalytic processes, otherwise it wouldn't happen. And this is how we define epigenetic inheritance. So the mechanisms must be in place. They must be involved in one way or another, just on logical grounds. So, and they do underlie the transgenerational transmission of acquired traits, but we have to put here a big and fat qualifier, because this would happen in different way, with different fidelity in different organisms, and also with very, very different effects. And it is our job as biologists and as people who are interested in theoretical biology and also philosophy of biology and history of biology to understand these things. And I think this is the way that we can make progress. So I just want to give you two examples. One is that it is not at all the case that epigenetic inheritance will lead necessarily to, we call it epigenetic mechanisms, will lead necessarily to similarity between parents and offspring. You can stress a parent and you can lead to compensatory effects in the offspring. And the effects in the offspring can lead to different effects in the grand offspring and so on. What is important is that you can have a cascade of changes that are going through generations. So when we're talking about transmission but through epigenetic mechanisms, it's not only similarity that we should be looking at. This is one thing, and there is a concept that was developed by a Russian biologist called epigenetic compensation, which is trying to give us this opposite side of the similarity coin. The other thing that we have to consider is what exactly is being affected during when we have this environmental change that changes some kind of epigenetic factor, and how is this then transmitted from generation to generation. So we can have a stimulus which affects the germline, right? The germline is the bit, the squares are organism, S is soma, and E and E1 are epigenetic changes in the germline over there. So we can have a soma, we can have a stimulus that is affecting the germline of the parent. The parent is not phenotypically affected at all, but the offspring is. And then this is transmitted through the germline, because it was the germline that was affected in the first place, and there are mechanisms which we have to understand in detail, and we do not, that are very important, that are operating from meiosis to meiosis. So we can have this kind of thing, and since we change the germline, the next generation will have a different phenotype. It will have phenotype S1. And we can have parallel induction. That is, we can affect the soma of the parent and its germline at the same time. And this can be inherited again through the germline. And we can have an effect which is called somatic induction, which we affect the soma. The soma affects the development of the germline, which is altered, and this can be transmitted, but the soma is changed, and the somatic change, let's say hormonal changes, can then affect the germline. So even when we see epigenetic inheritance in the germline, we should not be sure that it is that there are no somatic effects on it. I think we have to put the question forward, and I think that this way of thinking helps us to ask to ask these questions. When we think about it in these terms, I think this type of questions come to the fore. Now I want to finish with the implications of this thing. So, okay, fine, we know more or less what we're talking about. So what are the implications? So I think that for developmental biology, we need to study these processes of phenotypic accommodation. There is a very useful paper of somebody who presented the term adaptive improvisation. This adaptive improvisation processes that are very important for every organism, also the processes, of course, that are based on pre-existing programs, but also the improvisation processes. When we think about heredity, I think epigenetics points to the need to study the regulatory processes during mitosis, meiosis, and early embryogenesis that enable the maintenance of acquired variation over generations. Meiosis is an enormously important stage. It is a dramatic change. There are dramatic things happening there. Reduction divisions, huge changes in chromatin, resetting, everything. What is going on there? What are the rules? Can we find some kind of principles, rules, mechanisms that will enable us to understand how things are transmitted? And for evolutionary biology, and I want to say that even short-term inheritance can have very, very substantial effects, both at the level of population genetics and also to explain things like the missing heritability, the big hole of missing heritability. The fact that it is very, very difficult for us to understand why millions of QTLs don't manage to explain the inheritance of phenotypic variations in families and in lineages. For evolutionary biology, epigenetics points to the need to study many things. For example, we have to ask what are the origins of epigenetic inheritance? What did it evolve from? I mean, it didn't suddenly descend from heaven, right? So it evolved from something. What are the conditions that foster its inheritance? In what conditions is epigenetic inheritance selectively advantageous? It obviously is not in all conditions. It's a very bad idea to transmit all your bad habits, for example. And what are the various adaptive effects of epigenetic inheritance in different taxa? So I think there are many, many questions for us. There are many open questions, and we're in the beginning, I think, of a big intellectual adventure. But I think that to the question posed by this symposium, we can take the question mark out. Thank you very much.
Questions? Madame Junior.
Yes, I would like to make a comment concerning the differences between the animal models that we have in the lab and the human populations. Is that in the animal model, the F1, F2, F3 generations do not have changes in their conditions of breeding or whatever, what they eat, because we really strain to have exactly the same conditions. Whereas in humans, it's completely different, because a human can change his lifestyle, it can have different environmental factors, and therefore the response that the individual will have, depending on what he has been conditioned on in epigenetic changes, will be very different according to its sex, its age, and also the pathological evolution of what he had inherited before. And so I think it will be much more complex in humans, and that we should not expect to have exactly the same phenotype being transmitted from the F2 or F3 generation.
Yes, I agree with you. I think also with humans, obviously, we can't work with pure lines. It's not possible. So, and but I think that one of the things that we're learning is that sometimes to change an epigenetic mark in a way that is heritable, we need a big shock and a big environmental change. Because one of the things that about all animals is that they are highly canalized. In every sense, genetic variations usually make very little difference unless they are big enough. And the same would be true for epigenetic variations, and the same is true for environmental variations. So I think that one of the things that people are beginning to study, so people are interested in usually unfortunate kind of traumas, like looking at the people who were the victims of the genocide in Rwanda, and looking at the effects that this might have on their epigenotype, or Holocaust survivors, and all kinds of other horrible things that are so profoundly, that are the result of not only one-time effect, but a chronic effect for a long time, and they're trying to look at this. Now, as we have learned also, we are very lucky that there are experiments in mice that Isabel was talking about, and there are other experiments too by somebody called Arai, who are showing that enriched environment can alter this kind of things. So I think that we have to study the epigenetics of happiness as well as the epigenetics of disasters. It's very, very important to know how we can, but you're right, of course. So we usually will concentrate, I think people do concentrate in humans on extremely extreme kind of changes, environmental changes, or chronic environmental changes, trying to see what the effects are. But it's almost impossible to tease things apart.
I I had a comment and then a sort of more philosophical question, but the comment is, I really like the clarity with which you present the idea of replication, copying information, and reconstruction. I think that's very important, and these are concepts that I don't think everyone has completely taken on board, even though all of us working on epigenetics are actually realizing that it's about reconstruction. I guess my other point was about you have written a lot of books about epigenetics, epigenetic mechanisms, and you do touch on the cultural aspects and the transmission of information with culture. So I was just wondering whether you wanted to say a word as our last speaker about this.
Yeah, I think I think that when we're thinking about I think that we are a very interesting species. Here we are, sitting here. There are no chimpanzees here. And if they were, they would go crazy. They wouldn't have this patience to sit. And we transmit information through symbolic means, through language, and so on. And this is a major way in which we think, we represent things to ourselves, and in which we communicate. And it is a very important part of the way in which we construct our environment. Now, I think, what is the relationship between this and epigenetics? We know that changes in the cultural environment can lead to the selection of genes. So, for example, if we change the environment in such a way that suddenly people who are deaf and mute can communicate through sign language, because there are schools for people who can communicate in this way, we know from studies that were made in the United States that this would lead to a genetic change in the population, to the doubling of the frequency of homozygotes for the major gene for deafness, which is connexin deafness. Why? Because people tend to marry people with whom they communicate, and the people with whom they communicate will be carriers of this recessive gene, and therefore the frequency of homozygotes in the population will go up. So this is one very interesting example of the way in which culture changes genetics. There are several others. There's not a huge number of such examples, but there are several others. I'm giving this example because it's not the common example. The common example is the domestication of the cow and the lactase gene, which you probably all know. But if you don't, very briefly, I mean, with domesticated cows for milk 5,000 years ago, and the gene for breaking lactose, the sugar in the milk sugar, even after weaning, this mutation has increased in many populations which are using fresh milk. In populations which are not using fresh milk, we don't have the spread of this allele. Very, very interesting story, but it's another example of the way that we construct our environment through culture, through things like domestication, through building schools for the deaf, and so on, can change the frequency of our genes. It can also change the frequency of epigenetic variations, especially relatively stable, if there are some stable ones, or it can induce them even. Because there is no reason to think that a continuous kind of environmental change will not lead to changes in the expression of genes, and also some of them can lead to transgenerational effects. It's not terribly important in this case because the environment is perpetuated. The inducing environment is perpetuated. But of course this will happen. Everything will happen, and everything which is relevant will be selected, whether it is a mutation or an epimutation. I mean, natural selection really does not care. So,
Thank you. Thank you very much.